Human eyes, or in general, vertebrate eyes and vertebrate vision is extremely complex. And one could ask, where did eyes and vision come from? How might it have evolved? Well, before there were vertebrates, there were ancestral chordates by more than 500 million years ago. And one could ask, did they possess, complex lateral eyes capable of vision in the sense that we use with vertebrates? The answer is no. Did they, however, have the precursors of eyes? Did they have the fundamental components from which vertebrate eyes would be made? Yes. Let me explain. The core of vertebrate vision is this. There are proteins known as opsins, which associate with a chromophore known as retinol. Retinol will change its conformation based on whether light has struck it or not. After light strikes it, the chromophore retinol dissociates from opsin, and so therefore the association of opsin and retinol depends on light. And the cells which contain the opsin then signal neural tissue based on whether retinol is associated or not, so that the neural tissue can process the presence of light. This is the essence of vision. These pieces were in place in the ancestor of vertebrates. As I review elsewhere, opsins are G protein coupled receptors. These proteins are known in all living things. Opsins first evolved in bacteria. There are bacteria which respond to light. There are protists which respond to light. Responding to light is quite important, and it evolved early in the history of life. Opsin genes therefore duplicated, and there are many branches in the opsin gene family tree. The types of opsins found in vertebrates evolved over time. There are many of them, but the animal opsins are more similar to the vertebrate opsins than those of non-animals. The same is true of bilaterally symmetrical animals. By the time of chordates, ciliary opsins had evolved, which is the kind expressed on the photoreceptors of vertebrates. Where on the human photoreceptors, rods and cones, are opsin proteins expressed? They are on a modified primary cilium. In addition to cilia, which can occur in large numbers and help to, say, move the fluid around the cell, most cells have one single primary cilium. And this very often is modified to be sensory, as it is in rods and cones, where the opsins are expressed in the human eye. Cilia in general originated in the early eukaryotes, and most human cells possess a primary cilium. This can be modified for a sensory function, as it is in the visual cells, rods, and cones, where it is where the opsin molecules are located. The ancestral chordates, like the lancelets today, would have had multiple structures capable of perceiving vision, including the suspected homologs of vertebrate eyes, which possessed ciliary opsins expressed on the primary cilia. These are images of the spinal cord of the modern lancelet, a primitive chordate, and specifically areas which detect light. This sounds odd because we don't associate the retina of the eyes with the central nervous system, but this is a mistake. The retina is actually derived from the brain. It is part of the central nervous system. And thus, when we see in primitive chordates that the central nervous system detected light, this is actually linked to the development of light perception in the retina. If you look at how the retina develops in embryos, you can see it actually develops as part of the brain and then moves away from the remainder of it, probably because once the cartilage of a primitive skull began to develop, light perception was interfered with, and this photoreceptive area then had to be away from this developing skull. The optic nerve technically isn't a nerve, it's technically a tract which connects one part of the brain to another part, which then move during embryonic development. All vertebrates express opsins in their brain, and many detect light using their central nervous system. Even mammals express encephalopsin in their brains. This is most evident in the pineal glands, which is a part of the brain which in most vertebrates detects light and functions in circadian rhythms, adjusting the timing of biological rhythms to the day-night cycle. The photoreceptors of pineal glands are similar to and derive from a common ancestor with the rods of the retina. Even in the human brain, where the pineal gland does not detect light, there are these pinealocytes which are homologous to photoreceptors. Even the retina of the eye expresses opsin proteins which function in visual tasks other than vision. They are involved in circadian detection of light or visual reflexes. Many other genes link complex vertebrate eyes to those more primitive eyes of invertebrates. When invertebrate embryos are using homeodomain genes to signal embryonic cells to put an eye here, to differentiate photoreceptors in this region, they use genes which are homologous to the genes that vertebrate and human embryos use as well, such as those of the 6 and PAX gene families. The proteins of the lens, which are transparent and allow for the passage of light, are not unique to lenses. They are derived from other proteins which had other functions, such as heat shock proteins or enzymes. Ancestral chordates, like the primitive chordates today, did not possess complex lateral eyes capable of vision, nor would their simple brains be capable of processing vision. However, these chordates did possess the opsin proteins, the ciliary opsin proteins, and the modified cilium, which composes human photoreceptors, neural tissue capable of processing light, including the suspected homologs of the lateral eyes and or the pineal body, in addition to other structures which are the building blocks from which the vertebrates would develop their more complex eyes.